Aha, here we go. Great. Well, absolute pleasure to have you here, Bing, as well with Steve and Katie from Washington. As we've said before, we have a little systems neuroscience and complexity group that has really broad set of interests, and the name Brunton comes up more often than we'd care to admit. And I really was excited to meet both of you in a cosign and chat. And I have to say, we've had a couple chats now, and it always leads to really fun, interesting conclusions and kind of interesting corners of, I think, curiosity-driven science that I really like, and I think really resonates with the kind of work that we're trying to do here, and really different topics. And so I'm really excited that you were willing to come and chat with us and walk us through what I think is probably one of the more interesting stories that I've heard in the last little while about how on earth birds are able to balance. So without any further ado, I'll let you take it away, Bing. Thank you so much for joining, and I'm looking forward to the talk.
Thank you, Mac. Flattery will get you anywhere, or at least a seminar speaker. Okay, cool, everyone. So we're going to talk about this bird balance story today, and it's going to be very informal. And also, Katie and I are going to kind of do a tag team type thing. So we can talk about this for hours. But it will be much more fun if you actually ask us questions. So I think what we decided to do is I'm going to try to give a very short intro to the big picture project. And then Katie will take over and talk about more specifics. You are more than invited and welcome to ask questions and interrupt at any point. Please interrupt us. I know on Zoom, sometimes this is awkward, and because there are at least two of us, so like one of us will be able to type and stuff, and Katie and I don't mind interrupting each other. So if you type something in the chat, raise your hand, or if you if you just don't know when to unmute yourself, that's totally okay. We'll figure out a way to actually have this be interactive rather than us talking at you, because then you may as well watch us on YouTube. Okay? That sound good? Are we all on board? All right.
Yep.
Awesome.
I'm going to interrupt you and say yes.
Okay. Awesome. Appreciate it. Okay. So here, I'm going to share my screen, and hopefully you all see the slide. Does that work? Are you seeing the orange slide? Yep. Okay. So yep. I'll start all of this by saying that every single thing that we'll be talking about today has been done in collaboration with my collaborator, David Prikel. And all of it has also been led by our postdoc extraordinaire, Katie Stanchak. David is not here today, but Katie is. As some of you may know, I am entirely a computational neuroscientist. And so obviously none of the experiments that anything involving biological organisms is done in my lab. So all of it has been done in David's lab or with museum specimens that we have with other collaborators. So I just wanted to say that up front. So to Katie and I here are representing the project and also on behalf of David. Okay. All right, cool. And then, okay, so David is also doesn't actually study. He's like the previous big chunk of his career has mostly been in songbirds. And so he's an electrophysiologist, mostly working on the songbird circuit. And so he at least had birds in his lab that we really took advantage of. And so this was when this project started many years ago, it was a brand new thing for both David and myself. And so we were really excited to get into it. And so as Max said, it was like literally driven by curiosity. We heard about this thing. We could not stop talking about it every time we saw each other in the hallway, and eventually became a project. So here we go. All right, cool. So I'm going to start by showing you a couple of YouTube videos. Hopefully this plays okay over Zoom. We can probably post the links in the chat especially if your video is laggy on the other side, this may not work too well for you. So I'm just going to try it. Okay. So this first video is hopefully you're seeing a kingfisher perched on a twig.
Yeah, and it looks good to me. Yeah.
Perfect. Thank you. What you should be able to see is that it looks photoshopped. It looks like somebody made its head stay completely still, and the rest of its body is doing this crazy thing where it's going up and down and left and right, and occasionally it does make a small saccade with its head, but it's keeping its head very, very still, presumably to look for fish so they can go hunting. Okay. So this is done during perching. There's also very charismatic videos of this exact same head stabilization behavior during flight. And so here's a video that actually won one of the Nature Photography Awards a couple of years ago for the National Geographic of a hawk, a red hawk, who's stabilizing its head during flight. So it's hovering and there's pretty substantial winds. So it's, as you can see, moving every other part of its body except its head and looking downwards. Again, presumably because it's trying to find something to hunt and eat. Okay, so this is like exceptional balance and gaze stabilization behavior. Now, to be clear, most animals also do balance and gaze stabilization. You and I do it too. Or like otherwise, when you're on a walk, like Steve and Jordan are outside right now, their visual field would be completely uninterpretable if they didn't do some gaze stabilization and also some computation in the visual cortices as well. It's all we're saying is that the extent to which the performance envelopes of the avian species is pretty exceptional relative to other animals, including ourselves. This behavior has also been observed in invertebrate species, and so there's videos of flies also doing this like head stabilization thing because it also needs to keep its visual field still when it's flying and hunting. So the behavior is very general, but the extent to which the birds do this is pretty exceptional. It's so exceptional that as far as I'm aware, I'm about to show you the only car commercial I've ever heard of that does not feature a car. So here is a Mercedes-Benz commercial featuring the domestic chicken. So this is not like a weird hunting bird thing, because I showed you kind of two very charismatic exotic birds, a kingfisher and a hawk, right? But this is not like a weird bird thing. This is all birds. So here's the video. Okay, it's trying to advertise this really good suspension system for the new Mercedes-Benz, but really it's talking about how awesome chickens are and awesome birds are. Okay, so, and there's many, many more examples. If you have a chicken, if you have access to a chicken, you are encouraged to do this experiment by yourself. It's actually quite accessible to get them to do this. Okay, so that's the behavior we're trying to explain, and we're not the only people to have thought about this, but there was a crucial piece of information that when I heard of it totally blew my mind because I had no idea as a neuroscientist that it existed. And so we have a bird and it is balanced on a perch or might be in flight or something like that. Okay, so when we're thinking about balancing behavior in animals, one of the first things you think about in vertebrates is the inner ear vestibular system. So birds have an inner ear semicircular canal system just like we do. It looks quite similar. So we have 3 semicircular canals, fluid-filled bony cavities, right? And they're approximately orthogonal to each other. And this acts as a gyroscope inside their head. So it can detect angular acceleration in each of the 3 rotational axes. So they've got these in their heads, 2 of them, just like we do. And they look really similar and they function a similar way as well. We have the hair cells and the rest of it. It's extraordinarily well understood, very, very well studied system that we know about. And birds have them too. So your first thought might be, okay, so this has something to do with the inner ear. And that would be true, except if you thought about it, if they accomplish the task of keeping their head completely still, there is no acceleration signal in the inner ear, right? It can't be only the inner ear system, because if it were, like if the head's still, there's no signal. So it turns out that there is a very poorly studied alternative hypothesis. And so the next couple of slides, I'm going to go through the rough anatomical setup and tell you about what we know about the anatomy. Some of this is from a little bit of literature that we have we've found published, and a lot of the finer detailed stuff was work done by Katie in the last couple of years. So birds, as it turns out, They have a large bony structure called a synsacrum that is in their lower spine. So if you kind of like take your hand, you can kind of feel the last couple of segments of your spine in the lower lumbosacral region. You have these segments. Birds have them too. The exception is that those couple of pieces of the vertebrae have fused together into one gigantic bone. So instead of having separate bones like we do, it's fused together into one gigantic bone, and it's also fused with parts of the pelvic bones. And this is called the synsacrum. It's the single largest piece of bone that is in the bird. So if you get like a box of bones of a bird in a box, this is like the largest piece of bone that's in that box. Everything else is gone. You have a box of bones. This is the largest piece in there. If you look at the negative space inside the synsacrum, okay, we're going to do what's called a digital endocast. So when people used to do these, like when we're looking at the inner ear, it's an endocast in the sense that if you injected paint into the inner ear and then cracked the bones open and saw the paint that was left over, this would be the shape of it. People used to do this, and now we do it digitally. Exact same thing. If you like digitally injected paint into the hollow space inside this piece of bone, the synsacrum we're looking at here, and then cracked it open by doing a morphological scan, this is what the negative space on the inside looks like. And if you're thinking to yourself, that is how you're supposed to feel. So the most obvious thing about it is that it has these bony cavities that form canals. And there are several of them, and they are orthogonal to the axis of the spine, and they stick out. The second thing you might see that is weird about this negative cavity space inside the synsacrum is that there is a bulge in the middle where there's the diameter is larger here, above and beyond what the bony cavities that are sticking out, these canals. So we're going to call these vertebral canals, okay? And so there is a set of strong hypotheses, and we'll go through the anatomy and a little bit of the evidence in more detail a little bit later. But basically, if you even just look at this anatomy, the strong suggestion here, the hypothesis you might be maybe thinking to yourself is like, yeah, this could work like the inner ears, right? If these are fluid-filled canals, and there's some way of detecting fluid motion, then you should be able to detect acceleration, especially in the roll direction. And so here is a hypothesized secondary balance organ, basically in the animal's butt, close to its center of mass. And we believe that the coordinated function of this, what we call the LSO, the lumbosacral organ, and the inner ear vestibular system is responsible for the exceptional gaze stabilization and balance behavior we see in avian species. This hypothesis was first articulated by a German ornithologist, Reinhold Necker. Necker has retired. Necker has been retired for the last 10 years or so. I think we've had some contact with him, but we haven't talked to him extensively. But he was the first one to have actually articulated the hypothesis that we had just talked about. We all good so far?
So the hypothesis is that this is involved, but not precisely how.
Nobody knows how. This is almost entirely understudied. We have a lot of strong hypotheses that are basically very strongly suggested by the combination of the anatomy, some of the more evidence that we have found since, and also some behavior. Some of it we got from papers that Necker has written, and some of it we have reproduced ourselves. But like by the end of this talk, you will know as much as anyone knows about this organ. It's like, if you think about how much we know about other sensory systems, like how much we know about the retina, how much we know about the inner ear vestibular system, how much we know even about the olfactory system, the gustatory system, like this is almost entirely understudied. And this is the reason why David and I could not stop talking about it, because like as a neuroscientist and as somebody who cares about behavior, like it's not every day you hear about a brand new sensory system that you've never heard of, that nobody knows anything about, and is not like a weird esoteric thing in a strange species of bugs that you only find in the Amazon in one part. Like if we're right, this is in every single bird on Earth. Like all the birds that are flying around outside, all the crows that I've been feeding on my porch, they all have this, right? This is not a weird obscure thing. This is in all birds. Right. So the potential impact of this for understanding of not only balance and control, but bird evolution, I mean, it's pretty huge. If this thing is born out, right? But it's very, very understudied. We are one of only 2 groups that we're aware of who are currently studying this. And we know the other people who are doing complementary things.
So can I ask, Bing, then with this little endocast, how can we think about where the spinal cord would be relative to this, and or will that come out as we continue to chat today?
That is an excellent question. So I'm going to show you a movie that Katie had made showing this in a bit more detail. So I'm just going to let this play for a little bit. So here's the endocast in the same color as before. As you see the endocast disappear, you'll see the remainder of the soft tissue that is in this bird's spine. And so the spinal cord is kind of in that purple color, okay? The spinal cord is split open down the middle in this part of the spine. It's going to just play over and over because we're going to keep talking about this. It's split down the middle. I think Katie likes to describe it as being split open like a hot dog bun, which I think is a really lovely image. So it's kind of like you took the spinal cord, you cut it in half, and you split it open like a hot dog bun. And in the place of the hot dog, you have what's called a glycogen body, which I've never mentioned before, but it's another cool specialization of this part of the spine. The glycogen body is in there, and in the glycogen body is this ball of, I don't know, tissue. We can talk about the evolutionary and developmental origins we think of the glycogen body, but it's there. Okay, so let's just talk about the anatomy first, okay? It's there. It's inside the split open area. It's kind of like inside where the hot dog would be in the hot dog bun, okay? In addition, you can see by the way that the endocast is disappearing, that there is some unaccounted for space in the bony cavity, which we have, Katie again has very carefully checked that we are pretty sure that there was, like, it's not like there's shrinkage of the soft tissue or something. We think that that empty space is presumably filled with cerebrospinal fluid, just like it would be in the inner ear or semicircular canals. So
is the hot dog bun sliced in the dorsal or the ventral part of the spinal cord?
Go ahead, Katie.
Dorsal. So the ventral side's this blank purple face, and then the dorsal side, so where that expansion is in the endocast, that's where the glycogen body is.
Okay. And so going back to my in my sort of the corners, the dusty corners of the library of my brain from learning about neuroanatomy, that's the sensory part of the spinal cord, right? Where the dorsal roots come in, that area has been somehow kind of like split apart, and there's this weird, funny, sort of tumory looking thing sitting in that place.
So it's at the midline of the spinal cord. So the dorsal horns are intact, and they're to the sides of where the glycogen body is. Now, interestingly, the glycogen body itself is not just on top of the spinal cord, it's fully integrated in it. And I say this because the very bottom ventral aspect of the glycogen body surrounds the central canal of the spinal cord.
Oh, interesting.
The central canal goes through the glycogen body in this part of the spine.
Yes.
So is it like intermediate gray matter that's been expanded upon, or is it like, what is it cellularly?
So the glycogen body, it's hypothesized to derive from glial cells, and it has neurons running through it. We know this, but it's primarily these big fat cells full of glycogen. And just as a note, the accessory lobes are similar to the glycogen body.
Yeah.
So that's like, and there's more. Okay, so there's these pink things that are highlighted on this animation. And they are little pieces of tissue that are extending laterally from the spinal cord. We're actively pursuing more anatomical studies of these accessory lobes right now. What we do know is that their locations are coincident with the ends of the vertebral canals. So for every canal, there's a pair of them, one on each side.
So what, they're like at the end of the little columns that are running side to side. Okay, gotcha. Yeah,
they're at the ends of the canals. Mm-hmm.
Oh. So if you go up into the brainstem, I don't know about birds, but I'm assuming it's quite similar because it's pretty common across mammals. The kinds of cells that sit around the central canal of the spinal cord, this really fascinating class of cells that typically called the kind of like central gray matter, and they extend further up into the periaqueductal gray, which gets a ton of interesting press because it's involved in things like pain regulation and emotion and really quick fight or flight type responses. So this really kind of like core kind of elements of our response to the world around us, and it's also like woefully understudied. But there are a few studies that I know of that show like microscopically the cells there look really weird, very unlike other kinds of neurons that we typically see. And the weird thing about them is they're called allodendritic, and don't worry about the term, but it just basically means that they branch their dendrites in a way that they keep getting further and further apart. They get longer and longer branches rather than shorter and shorter branches. So if it was a tree, it would look really weird because the leaves would be like way far away from the bough, and the bough would be way far away, but less so than from the trunk, right? And so as opposed to what you're used to seeing, which is that the distance gets smaller and smaller. But then when they branch out and do this weird thing, what they're doing is they're like synapsing onto often white matter tracks that are running up and down the spinal cord and the brainstem from heaps and heaps of different systems. And no one's really kind of figured out why you would organize that system that way. But it turns out it's incredibly important for like just being awake, like arousal. If you knock these things out, you typically have a coma or die. And so they're incredibly important for like general neural function, but really understudied and really weird looking. And so I'm very curious whether or not this little glycogen body you see has some of these guys in there, some of these kinds of weird allodendritic cells. I can send you papers later, but I'm just a curious aside.
Yes, please. Yeah, that's an interesting. I don't think I've never heard of that connection before.
Yeah, the one guy that studied them has written a book about them. He's actually got a comms paper about some of the genetics of these cells. But it's a weird quirk of neurology history that there was a big competition early on between the sort of ascending arousal system way of thinking about a brain. Maruzzi and Magoon had this really beautiful work showing that if you stimulated parts of the brainstem, you could cause people to have seizures. A guy called Rudolf Hess won the Nobel Prize in the 40s. We were actually talking about this in lab meeting yesterday, for stimulating parts of the hypothalamus, which is a kind of extension of the arousal system, and causing animals to become really angry or go to sleep. There's this really interesting kind of rules of kind of like neural engagement baked into this circuitry. But right around the same time, Sherrington was up in the brain of humans that were having surgery, and he was stimulating parts of their brain. And he was saying, oh, if I stimulate here, the person sees a flash of light. And if I stimulate here, they move their left arm. And if I stimulate here, they hear a tone, right? And so the entire field moved towards this idea of localization of function up into the cerebral cortex, and they just left all the other stuff behind. And so the entire funding scheme, everything went to the other way. And part of what we're thinking is fascinating at the moment is just like trying to bring the attention of neuroscience back to this more integrated whole brain set of answers for how a brain works. That incorporate all the other stuff. So anyway, it's super understudied, but I'd be happy to pass you that stuff. I don't want to distract from the main message.
No, that's really interesting. Yeah, we have been really interested in various aspects of what these cells actually are. What do they look like? I mean, the number of questions far exceed the number of answers at this point. Okay, but what we do know, here's back to what we do know. We're fairly confident in saying that all living birds have one of these structures. And so in Katie's paper from a couple of years ago, done in collaboration with Cooper French, who is an ornithologist graduate student, we basically went to our local Natural History Museum, the Burke Museum of Natural History, and took a bunch of birds they had and scanned them. And so Katie and Cooper scanned all these birds and looked at the digital endocasts of their bones. And in every single bird species we looked at, they had these vertebral canals in the digital endocast. And so the representation here, you can kind of see that we sampled all the birds that currently live on Earth, and they all have one. We have then looked at lots of other vertebrate species, and sadly, none of the rest of us have one. So we, monkeys, mice, bats, lizards, fish. This is Nobody else has one. So that was very sad. But this is a bird thing. Is that an accurate representation of the result, Katie?
Yes, although I'd say that there's some interesting cellular level analogies, if not homologies, but definitely like this whole crazy morphology of the spinal cord is only a bird thing.
Yes. Secondarily, Katie and Cooper then did an analysis comparing the prominence of these canals, because in some species they're gigantic, they're sticking out a lot. And in some species, the whole thing is like barely there. It's kind of a tiny little bump. And so we quantified the prominence of these transverse canals. And there was a relationship, there was a trend for birds that have a perching lifestyle with larger canals. Katie's in the middle of making this analysis more statistically rigorous with a larger sample size. And so this first study, I think we had like ballpark 50 bird species. And we now have more, and we can do a more rigorous morphological analysis. And so that's in the works.
So if we look at a paper that I know that we both really admire from Paul Chisek, sorry, I always get his name, I always pronounce it wrong, the phylogenetic refinement. If you look at the little cladistics tree he has, the nearest neighbor for a bird is a dinosaur. Do dinosaurs have something like this? Is it possible to look at, the big T-Rex? Can you go like with a...
Excellent question. Katie, you want to take this one?
Yeah, it's a great question. It's something I've, really wanted to do for a long time now. Definitely. So I think the first step, what we'll see it would actually be in the literature. Something I've had kind of on the back burner is just figuring out the evolution of the synsacrum. When all these vertebrae become fused, because that is kind of required for the LSO to be there. And it starts to happen right as you get to the lineage to birds. Now, but there is, however, so back in the day, there used to be this really interesting hypothesis. I don't know if you've ever heard of it. It's called the dinosaur hindbrain hypothesis. So say a stegosaur has in its sacrum, which is fairly long, this big expansion. You can actually see an endocast that I think Cope took at Yale. It's like this big expansion in there. And basically the idea was that because a stegosaur's head is so small, like there must be more brain somewhere else, and like maybe it's back there. So nobody thinks this is true anymore, right? Like that's fine. We all know that like you don't need to have a big brain to be able to survive, and maybe it wasn't that smart. Who knows? Who cares? So it's now thought by the people who have noticed this that maybe that was, a glycogen body. What's super interesting about this is that the dinosaurs that have that expansion, so the stegosaurs, are not on the branch of Dinosauria that leads to birds. And that when you get to like the raptors and so on, they don't have that. In fact, you have like a reduction in the width of the spinal column. Obviously, at some point, there's another expansion into birds, which would be very interesting to know when that happens. Side note, there's like the crown line birds that survived the extinction. There are other branches of the bird tree that did not survive the extinction that are different. And it would be interesting to look at them too, including like perching birds and stuff that are just like, like a totally different group from the ones that survived, the enantiorniths. So there's a lot of really
What about
interesting...
pterodactyls, Katie?
Pterodactyls do not have this in the ones that I've looked at. There was actually a paper published on a few pterodactyl sacra a few years ago now, and they published their CT scans that they had too. And there was no evidence of this sort of thing. But that's a good question, yeah, because pterodactyls would also be, so dinosaurs are the most closely related to birds, right? But then you on the other lines, you have pterodactyls and crocodiles.
Yeah. I was trying to think of other animals that we call flying, but they're probably better described as gliding. Like flying lizards have like webbing in their between their arms and their legs, so they can kind of glide down from the tops of trees. And same with flying foxes and things in Australia, but they probably don't have the same to like sit and like perch and keep part of their body moving while they focus or something. That may not
Yeah. So bats definitely don't have this sort of thing. And then as
Katie
for,
did
and then
on bats, so she was really sad.
yeah. And then there are, you're right, there are no, birds are really the only flying lizards. So yeah, the rest are really gliders. So yeah,
fascinating.
although, yeah, I will say that there are like, it is interesting on the cellular level with what's getting to be known about that, how mechanosensory the spinal cord is. I think there's a lot of work in that area where this bird LSO is really, could really be like an adaptation of some neural architecture that's common to all vertebrates. But it's still true that birds are the only ones with the LSO and like the glycogen body and the accessory lobes and so on.
Yes, we'd love to know more about dinosaurs.
Yes.
Katie's been trying.
Yeah, I've visited a few museums to look at stuff. I've like, picked out like specimens and things that I would like to scan, but it does, I mean, it's kind of a project, right? It's not like, a lot of the birds we looked at, there's, maybe hundreds of them in museums. They're very common. That is not true of dinosaur fossils. So it's a real project to pack it up, get it to a CT scanner, right, that I can't just go in and like borrow one, although I do have a couple, but
I was thinking more nefariously,
harder.
you could like sneak up to a archaeological dig, and then as they're like pulling the bone out, you like run up and grab it and quickly look at it and then just run off. Right?
So
you wouldn't need to see it
have you ever heard of the bone wars back in the day? So people used to like do that. Paleontology,
they would like switch them out and stuff, right? Yeah.
Oh, they would like it was violent. They would like steal things from each other, and yeah, it was pretty bad. Hold each other at like knife and gunpoint and steal fossils from other groups.
Yeah, it's pretty gnarly.
Anyway, yeah, that's no longer like that in most cases. As far
you don't want to rekindle that fire.
No, I don't think that's
opposite.
Probably
of
not a good
science.
way of. Yeah, collaboration. I mostly meant it's kind of expensive. You have to get people at the museum to like sign up for this. And when I was really starting to do it is when COVID hit, and then it's been hard to catch back up with all the other stuff we've been working on.
Okay, so as Katie already started alluding to, there are a lot of different aspects of understanding what the LSO is doing. And because nobody has done any of this hardly, we have aspirations of answering this question of what is it, how does it work at a mechanistic level at each of these levels of description. So we showed you a little bit of the results from an evolutionary comparative work, looking at different extant bird species. Katie's already talked about some of the paleo aspirations. We want to look at it at the level of the organism and behavior. We want to understand more about the anatomy. We want to understand something about the neural circuits. What are these cells and who are they connected to? Nobody really knows. And what we're going to go into now is Katie's next paper that came out last year, giving us a really interesting clue about how this might work at the level of cells, synapses, and molecules. So it's another like really intriguing set of results that complements the gross anatomical descriptions that we had just gone over. And so the idea that there might be mechanosensation inside the spine is not new. So there is I would say better studied community, like a larger community of people who have done this, looking at mechanosensation of cells that are inside the vertebrate spine. A good fraction of this work has been done in fish. I don't know why, that's just where they've done it. And so, for example, Claire Wyatt's group in Paris has been studying what are called cerebral spinal fluid contacting cells, CSFCNs. These are mechanosensitive cells that are actually facing in, like they surround the central canal. In fish, we've known about these for a long time, and all other vertebrates have them too, and that's been known since going back to over 100 years ago. And then more recently, there was another paper that came out from Picton in 2021 showing that there is a spinal proprioceptive organ that is responsible for different, for sensing of the bending of the spine in fish. And there they actually worked out that these cells were glycinergic neurons and a little bit of the neural circuitry involved in how they're connected to the rest of the sensory motor control system. And so we have been in contact with both of these groups and have taken a lot of inspiration from their results. And so taken together with this, I'm going to show you the kind of the high level picture of what Katie has found in her next paper. So I'm just going to show it in one slide, and she's going to tell me where I've gotten it all wrong and what actually happened. Okay, so the biggest thing that you may have noticed is that because everyone's been studying fish and it's all about spinal bending, the biggest thing, the most obvious thing about the bird system we've been looking at is that because whatever is happening in the bird, the syncytium is definitely not bending. It's this one gigantic piece of bone. It's hard. It's not bending. Whatever it's doing, it's not bending. Okay. But there is definitely precedence for mechanosensitive cells being found in the vertebrate spinal column. But because it's not bending, is what we have found, what Katie did find, is that there are molecular markers of molecules that are associated with mechanosensation that we have found in the spine that have never been found in the spine before. So in particular, if you don't know this I had no idea about this either. I've been learning a lot of it from Katie and from David. For example, there is a molecule called myosin 7A, and it is associated with the inner ear balance organ and is in fact considered a molecular marker of inner ear hair cells. It's never been found in the spine before, but here they are in the LSO region of the bird's spine. And there's a couple of other ones. I will leave Katie to talk to you about all the detailed molecular findings of what we found, but I will also say that one of the reasons we're really excited about this is because having a couple of molecules that we know to be expressed specifically in this region also gives us a viable target for future genetic manipulations. So it is possible to do genetic manipulations, like do the kind of the dream opto experiment in the bird spine. But that requires having a lever into the system. And so Myo7A and perhaps one of these other molecules might be that. Okay, so that's the intro. I'm going to let Katie talk about the more specifics, or like people can ask questions here if they want to. So this is getting really molecular now.
And I'll just pop in to say, like, I think they're doing it in zebrafish because larval zebrafish are basically transparent, so you can see everything. And it's unfortunate they don't have an LSO because the bird LSO is encased in bone, which results in a lot of practical problems. But alas. Does anyone have any questions?
We don't ask these questions because they're convenient.
No. Okay. So should I share my screen and kind of expand upon some of these molecular results and how they fit into our hypotheses for how the LSO might function?
Does that sound good, everyone?
Okay. I think I'm going to, okay. I have one screen here, so I'm probably going to lose. Oh, I can still see everybody. Great. Okay, so just to, because it's kind of important, I'm going to reiterate some of what Bing said about what's already known. So these cerebrospinal fluid contacting neurons are one set of known spinal mechanosensors, and they are located in the central canal of the spinal cord. And the idea is that they detect flow through the cerebrospinal fluid in the central canal. So you can imagine if the spinal cord bends, you might get different flow patterns, and then activate these cerebrospinal fluid contacting neurons. And the other is the Picton paper being mentioned, these lateral cells that detect bending and strain. This is piezo 2 activated, and they project then contralaterally and are involved in this sort of centralized sensory motor network or localized sensory motor network. And importantly, they're these glycinergic neurons. and work through this network via their inhibitory nature. Okay, so through all this conversation and reading a lot of the literature of what other people have published before, we came up with a set of hypotheses, non-exclusive hypotheses. So there could be multiple of these acting at once for how the lumbosacral organ might sense. So the first is the one that Necker, who being mentioned before, had come up with, and it's the idea that you could have cerebrospinal fluid flow through these canals that then impacts the neurons, say putative mechanosensory neurons in the accessory lobes, because the accessory lobes line up with these canal-like structures. Another is that this whole structure is suspended on a set of modified spinal ligaments. So it's a modified pia mater, actually, I believe. And so the idea is that the neural, because you have these interface of two very different tissue types, you could get strain there as the whole spinal structure, the whole neural tissue structure is suspended and thus could jiggle. We call this kind of our jiggle hypothesis. One of the, so you could get mechanosensory information there. The next is that you also have two different tissue interfaces between the glycogen body and the neural tissue. So a similar thing could be happening there. And then of course, the central canal, because we know that there are mechanosensory neurons associated with the central canal already. So this is our set of hypotheses that we're working with at the moment.
Okay, the accessory lobes, are they a sensory structure or a motor structure? Like, how should we think about what they're doing?
They're made of the same thing as the glycogen body, and they're hypothesized to be a sensory structure, but it's not known. So it is known that the neurons in the accessory lobes, one thing Necker found, they do project contralaterally. So that's something that is two motor neurons. So that's one clue that they might be sensory and not motor in particular. So
Thanks.
it's a lot of hypotheses to work with, and doing physiological experiments is really tricky because of the way the structures encased and all that, although we're definitely heading in that direction. But the first thing we want to do is kind of nail down some of these hypotheses by figuring out what are the tissue and cellular level characteristics of the lumbosacral organ. You know, where are cells that we might be able to target in some way. So that's where we started these molecular studies. So the first is for myosin 7A, and it's actually essential for hair cell function in the inner ears. And we do find it in the lumbosacral organ, and Bing showed a few samples of this, but here are some more details. And I have right here this cross section so you can kind of see where we are in the anatomy of the lumbosacral organ. And we find myosin 7A in the accessory lobes, as well as at the interface of the lower corners, the ventral corners of the spinal cord and that spinal ligament. And here's a close-up of what that looks like, the endings of those labeled neurons look like. These are immunohistochemistry experiments with that ligament. We also find espin, which is an actin bundling protein known to be involved in sensory neurons. And we find that in the accessory lobes in a weird formation with the myosin 7A, which doesn't make a lot of sense. And this has led to one of our next study to try to get some ultrastructural information about these accessory lobes and some better imaging, higher resolution imaging of what these myosin 7A fibers and espin marked fibers might actually look like. We also find myosin 7A in these cells that seem kind of affiliated with the central canal, although they're not in the central canal themselves. So it's these big cell bodies that are right outside the glycogen body. And so we look at this colored cross-sectional image, where we are is around here. And the central canal goes just through the bottom of the glycogen body. So you always see the glycogen body surrounding the central canal, not by much, but it definitely does. And then these are the ependymal cells that surround the central canal. And they're all, we find within the LSO these myosin 7A labeled kind of large cell bodies near that area. Okay, and then because of this Picton paper, we also thought, okay, well, we'd look for glycinergic neurons in the LSO. And we find that those myosin 7A fibers that we find in the accessory lobes and against the ligament in the lower ventral corner of the spinal cord, they're also glycinergic. And those myosin 7A cells that are really, that seems to be somewhat affiliated with the center of the spinal cord are glycinergic as well. And we haven't characterized this, but we have several sections that show glycinergic fibers affiliated with the central canal and ependymal cells themselves. What we don't know is where that, we haven't done any neuronal tracing, so we don't know where those come from. We don't know if these cells are affiliated with what's going on at the central canal. But those are all like interesting questions we're hoping to answer at some point. And then I'll also show a result for calcitonin gene-related peptides. So we did some studies with this because it's found in sensory afferents in the dorsal horn. So it's involved in sensing pain at the periphery. And so it's found in these nociceptor afferents they have their cell bodies in the dorsal root ganglia of the major roots that enter the spinal cord, and they enter through the dorsal horn. So this is like what you were referring to earlier, Mac. And this is what we find in the LSO. So we have a ton of labeling in the dorsal horn of the spinal cord, like you'd expect. But we also find CGRP labeling in fibers that cross the glycogen body, the midline of the spinal cord through the glycogen body, which is really weird because normally sensory afferents do not, they do not cross the midline. They interface in the dorsal horn with interneurons that then go and do, other stuff. So in a nutshell, if we come back to these hypotheses, what we found is kind of a little bit of molecular evidence supporting the involvement of all of this in some sort of sensory function. So the glycogen body with those CGRP labeled fibers running through. They're also substance P labeled. I didn't show that, which is another sensory peptide. We do see cells around the central canal area of the spinal cord. We have myosin 7A labeling both in those cells around the central canal, as well as in the accessory lobe and at this interface between the dentate ligament and the spinal cord. So this gives us a lot of, even though we haven't, narrowed down the number of these hypotheses, it gives us a lot of more specific things to look at. And then, does anyone have any questions yet?
I guess I was wondering if it's like a control problem and you're imagining the bird gets the signal from here, like this is its sensor. Is there any asymmetry in the precision of the 3 rotational directions or something like that, that the bird is kind of better at this direction than that? And that could match and give evidence for one or more of these hypotheses that might make the sensor more sensitive to one rotational direction over another.
That's interesting. Yeah, I wonder if there's a way to devise, a behavioral experiment with birds, where if you rotate them, hold them in like a harness or something and rotate them in different directions, when are they most able to
Correct.
focus their head?
Right.
That's interesting.
You could probably glean it from behavioral videos. Like you could just match the skeleton to some sort of wire framework, track the rotations, and then look at the head and see when the head has to make a compensatory movement when the angle is off axis, like Ben says. And that would be evidence for this as well.
Yeah, that's a good idea. Because there are yeah.
Do you
But
have
could
a
you
picture?
match-
Do you have a picture of the instrumented perch, or do you want me to show mine?
You could show a picture of it. I just have a video of a bird on a perch. It doesn't look like anything special. Do you have a diagram or a?
I have I mean, could you? I don't think I can share my screen as long as you're sharing
Oh.
yours.
Yeah, I can stop share.
This is all I have for now. It's just a schematic.
Oh
that I
yeah,
got.
this is what I was gonna pull up. Yeah,
Okay, perfect. Same thing.
this is great.
Katie's a wizard and does everything. So this is another set of experiments at the behavioral level that are ongoing. So this stuff is in progress, right? But the vision here is to really look at exactly the types of behavior that Ben just alluded to. So we're trying to build something that will allow us to be able to do those experiments as you propose, Ben, to do these targeted perturbations in different axes of rotation. And so Katie, along with Eddie Lumsden, and there's a couple of other people who have contributed to various parts of this effort along over the years. We're trying to build a instrumented perch for the bird to sit on that has the ability to measure forces, but then also apply torques in different directions. At the same time, we would like to do high-speed videography in 3D so we can track the kinematics of the bird as they're perching. And then they're also going to wear an adorable little pink backpack and have EMG wires. And then like this is just again, like this is the status of, this is not working as a integrated system yet, but this is kind of where Katie is headed with a different set of collaborators. Sorry, Katie, I know you had the same diagram.
Oh, no, it was actually, I would have had to like look for it. That wasn't in my presentation. So this was good. Yeah. Yeah. So the idea is that we'll be able to get applied force through the EMG. We can kind of figure out how they're using their muscles in response. And then the perch will measure a torque. And then we had an old perch that would measure angular rotation. But now the next iteration will have a cute little force sensor applied to it. And yeah, and then we're kind of at the stage where we don't have EMG working in a behaving bird, just in an anesthetized one. It's a little tricky because these zebra finches are small, so we need small electrodes.
But
Yeah,
I
so
like
that's
your
the general idea.
for asymmetries in the behavior axes of perturbation. That's a good idea.
Is it worth noting that this is potentially it's not the only system that the bird's using for balancing? Like I know, for example, cats have incredible head stabilization. They're not to the level of a bird, but there's plenty of videos where you can like move the cat's body around like you showed with the bird and it maintains it. And presumably it doesn't have this organ. So like there's a level of proficiency that can be gained with small recordings, presumably from this in ear circuit. So you kind of have to tease those two components apart.
Absolutely. In addition,
Yep.
there is extensive proprioception throughout the body. Right?
Yep, so it's, yeah,
Yes.
balance comes from eyes, ears, proprioception. Sure.
Is it
Yeah,
possible
it is tricky.
to block their ear, or is it cruel to? Could you somehow block the ear signal and isolate just the?
Are you proposing blinding the birds, Ben? Is that what you're proposing? You are not passing ethics at all.
Especially with the little pink backpack on, I don't want to cause it any harm.
I know. So we can turn the lights off. So that's fairly, it's easy enough with the eyes. There are ways of killing the hair cells in the inner ear. It involves a really high dose of antibiotic, which is how most people kill them, and it works, but it can have other effects too, as you might imagine. The bird gets pretty sick. So it's a tricky experiment.
The dream experiment would be a targeted, temporally precise manipulation at just the LSO. We want to be able to turn it on and off quickly and have it be completely reversible.
Yep. So we've had a couple ideas. One is like an optochemogenetic approach. We could get there. There's obviously that is non-trivial in birds, but that would be great. Another idea that we've had is like, if we could find a way to like implant a little magnet in the glycogen body, and then, you know, stick an EM coil around it, and then we could maybe turn on and off and like get it to twist to one side or another in response. I don't know. So, but that's an idea that has come up too.
I had a question about maybe one other way that this might work, that I think maybe I thought about this after our lunch, just because thinking about the kinds of things that Steve's interested in, was whether or not the different viscosity of CSF versus whatever is in the glycosynergic body might allow some kind of boundary friction or something like that the animal could sense. I don't know if you could think about how to test that, or Steve might have some thoughts about that as well.
That's interesting.
That's actually, Katie, this is the kind of thing that Ed studies, by the way. Right? Like,
yeah, so my understanding of glycogen, right, is that it's like this big clump of glucose that was really, really good at trapping water, right? And so you get like these little clumps of sugar that have water all around them. So it's extremely aqueous. And I looked it up, and I have no idea what a Pascal second is, but water and CSF have different Pascal seconds. So there's a gradient potentially between this like CSF bath and this really densely water-packed thing. And I wonder whether or not if you have those mechanosensors, they could kind of figure out the difference between that viscosity as it kind of moves, like, because the whole animal's body changes, right? As Preetham said in the comments, the fluid is going to shift, and the fluid of a particular density will shift faster than another one of a different density. And yeah, I don't know. It just seemed like that might be an interesting signal for the bird to pick up on.
That's an excellent suggestion, Mac. And we we have had similar thoughts with respect to differential density being the
cool
mechanism. Katie has also, we have a different collaborator who has just started. We got them interested enough that they're going to help us do some physical experiments.
Yeah, sick. It's kind of analogous a little bit. Eli and I were talking yesterday about the Wright brothers, because I've read this interesting article about how they solved flight by using their background in bicycle maintenance rather than trying to engineer the lift. You engineer the balance. And I was thinking about this as like a kind of wind tunnel type experiment, right? Where you get like two different density materials and sort of whoosh them against one another and figure out how big of a gradient you need in order for the barrier to notice the difference in movement of a certain. There must be some, biophysical law there of how these things work. Could have a lot of fun with that.
That'd be cool.
Yeah, that's interesting. Yeah, that's a good thought. We've been thinking of this in the sense of like that they have different densities, they're made of different materials, and they'll have different vibrational frequencies and things like that. And like that, it could get picked up by that. But I hadn't thought of the boundary contact between the fluid and the the first thing that makes me wonder is like, well, are there any mechanosensory neurons at the glycogen body that would pick that up at the surface?
100%,
but at the
100%.
same time, if it just causes a force at that boundary and it causes the whole thing, that could be what causes it to jiggle, right? So yeah. Which
it's funny. I love this idea of kind of natural selection being a little bit like water sort of falling into the cracks on pavement, and that if there's an opportunity to be taken advantage of, it'll kind of figure it out. And this seemed like a really interesting way to like take something that's already there in a system, like the amount of glycogen you have in your spinal cord, that just so happens to kind of give you this accident of having it rub up against CSF to give you that information you need. And then you just elaborate on it. An animal with a bigger, bulkier one, the one with a more fused sensacrum, would just benefit more because it's got a stronger, more robust signal to tap into. So it seemed like a kind of fun example of that. Maybe it's just a hypothesis, right? I mean,
no, it totally is.
Yeah. No, it's fascinating.
I think one of the joys and frustrations of working in biology is that when you have hypotheses, the answer is almost always all of the above.
Yeah,
like,
exactly.
You write these papers pretending that it's going to be alternative A or B, but at the end of the day, it's like, yes, it's those things.
yeah. I kind of love that part of it. The answers are just different flavor, right? than in physics, where the answer is like an equation with a nice little exponent on
Well,
it.
the answer in physics is usually 0 or 1. And if it's not, we just do a normalization and we're all done.
There's a question in the chat that there would only be fluid movement when the whole body is moved. And when that part of the body is moved, right? So you keep the head still, but yeah, when the whole like sensacrum lower part of the body, because it's all fused, it means like the pelvis would also be moving. Yeah, there's no CSF pump, right? So is there in the brain? I don't think so.
Is there a what?
CSF pump?
Oh, the CSF is made by a weird, mysterious little organ, or like a little clump of cells, called the choroid plexus, right in the ventricles, and it sort of seeps it out. But I don't think it puts it under pressure. But I do know that there are massive problems if the pressure changes. So, like if you pinch the spinal cord, you can cause you can like cause people to die from hydrocephalus and things. And so it's definitely in a range where if it's too low or too high, you're in big trouble. So I don't know if it's actively maintained at that, or if it's just like the physiological constraints of like having a spinal cord of a particular density, of a particular length, means that you keep it at a particular pressure.
Oh, that's a really good question, because I think there would only be fluid movement when the body moves. But if there's, I mean, there might be like absorption and then secretion, but I don't know if there's any flow causing mechanism. I don't think there is, but I don't.
In the ventricles, there are. So in the ventricles in the brain, there are ciliated cells that are line the walls, and they go like, right?
Really?
Yeah.
Oh,
so
and so they do.
Yeah.
It's a mixer.
So like it's not like a heart is a pump, right? Like there's not a pump like that I've heard of, but I have heard of ventricular circulation.
Like a kelp forest.
Exactly. Yeah. I like, but the other way, like it's ciliated movement, then like kind of moving the fluid, you know?
Yeah. Ooh, that's cool.
Like
so
whether or not that is enough to cause a pressure change, I doubt it, but I don't know.
Yeah, I feel like that wouldn't have a big effect on the spinal cord, but I feel like it's not very scientific. I have no idea.
Yeah, I don't know.
Yeah.
So I have a silly question. When people sleep, the ventricular system changes substantially. Their cells kind of like shrink down and the ventricular system expands, and all the CSF starts flooding into the spaces between cells. And the thought is that it's somehow to like clean out all the crap from the work that you've been doing during the day.
The lymphatic system.
Exactly, yeah. And so my question is, when birds sleep, do they sleep perched on a tree and still need to be fulfilling this balance problem? Or is it kind of less important because they're in the bough of a tree or something, and it's like, or the trunk or something, yeah.
Some birds do sleep while perching. One way they help with this is that they sit kind of back on their legs. So they're not like standing up necessarily, but it's still a complicated. Yeah, but it's still like they still are balancing while they're sleeping. Like it's still in a state where they would fall over. They're still upright.
So sleep on one foot, right?
Yeah. So there's a great example. There has been a hypothesis. This is interesting, because there has been a hypothesis that the glycogen body, back up a second, we know it's not used for energy storage, mostly from a bunch of old experiments that if you starve birds, it doesn't get smaller. And it doesn't have one of the enzymes required for, I didn't study
glycogen, like
biochemistry.
glycogenase or something.
Yeah, it's like glucose 6 phosphatase or whatever, one of the conversions. So that glycogen doesn't get converted to glucose in the glycogen body anyway. But there has been a hypothesis that it could be used to as a secretory organ, or so chemically managed through the central canal, parts of the nervous system, or as a storage, because the glycogen could still be converted to fats, right? So for say like myelination and stuff like that. I don't know how that would work, but that's
what about
another
the packing
hypothesis.
peanuts hypothesis? that it's just there to kind of buffer movement, because the really key stuff you need to keep your nervous system alive is sitting right in the core. And the last thing you'd want is like some mechanical object to come in and like tear that apart. So just put in a bunch of water around you, keep it stored, and then you can basically buffer movements. Something super boring like that.
Like how they build large buildings in earthquake areas by having a counterbalance.
Is that how they do it? Oh, really?
There's a
There's
counterweight.
counterbalances.
Yeah, there's a counterbalance at the bottom of the building in Tokyo.
Oh, sick. Yeah.
So is the idea
Engineering's
here that
fascinating.
the CSF around all this neural tissue would be kind of acting as a buffer itself?
Yeah. And then, I mean, I was just sort of thinking that the glycine bodies, if they're not being used for energy, are just little like water, like what are those water beads that people like on YouTube throw into pools, and they fill out the entire pool, and then they jump into the pool and it's like stuck in the, my kids had these for a while when they were younger. Have you guys seen those things before? It was like, oh, they're like, they're fun little things. They're these water, they're little beads, and they soak up to a certain density, right? They get to like,
Oh, do they get optically transparent? I've seen those. Is that what you're talking
yeah,
about?
exactly.
Oh
And then people will like jump into a pool.
Water, so they look invisible, but they're there.
Yes, exactly.
I've never had a pool of those. That sounds amazing. I've had like a small jar of them, and they were pretty cool.
Yeah, but so imagine like if you were like putting your delicate brainstem right in the middle of that, it's going to move much less and get perturbed much less if like all the water was just sort of sitting around it. I don't know. It's just a harebrained thought.
Oh, so you mean that like the spinal cord ends up suspended, maybe for a sensory reason or whatever, and then you end up with all this space, and it's like, whoa, too much. Let's just put some glycogen cells in there.
Yeah, just
like,
to buffer things.
Yeah.
Like a stupid, boring mechanical explanation for the exquisiteness of our nervous system. I kind of like it.
It's
maybe I feel like
isn't that wild that there's this whole gigantic structure in birds that we have no idea what it actually does.
Oh, I love it. It's so good.
that still indicates that there's something important about this part of the spinal cord, though.
Oh, absolutely.
Yeah, like why is not every other part of the spinal cord being so buffered?
Yeah. Yeah, 100%. I think so. The flip side of that comment you made before, being about how biology often has the answer of all of the above, is that it's also kind of all of there's heaps of answers that are really satisfying in a way, depending on how you look at it. I quite like that aspect of it. And yeah, that this could be it, right? Like that you have this thing that was around and it gets expanded, and if you expand it, then the animal that gets it gets lots of benefits. And some of them are balance, and some of them are, resilience to damage, and some of them are some other thing. And I actually really love these kind of like exaptations and kind of Waddingtonian answers for biological adaptation, just because it's like, who the fuck knows what the answer is going to be in the future? Who knows what the question's even going to be? You have to prepare with variation, and then later on the opportunity arises and you take advantage of it. And I don't know, I like those kinds of aspects of biology a lot.
Yeah, no, me too, for sure. I'm a big fan of these types of weird anatomical things that pop up for some reason or another, or not, right? And yeah, for sure.
Sorry,
So
is the size of the glycogen body like stable? Does it increase or decrease seasonally or anything? It just stays the same.
I don't know about seasonally. So there have been studies like starvation studies done on chickens and stuff like way in the past. I mean, they're fine studies, and it doesn't change in size, at least when you starve birds. I know that. Seasonal is an interesting question. I don't know. I'm
not aware of having come across that result one way or the other.
No, I don't know if anyone studied it. I will say I did another project in the Perkel lab. I was looking at seasonality in sparrows, and I've dissected a few of them, including some that have been hormonally manipulated to be at different seasons. And I didn't measure anything, of course, because I didn't think about it, but I certainly didn't notice any difference.
And are those the sparrows where there's pretty large changes in the nervous system with respect to seasonality?
Oh yeah.
Because that's why Perkel studies them, right?
In the song system, the males have huge changes.
Yeah, there's like huge changes with seasonality, so it's at least not noticeable on that order of magnitude.
Yeah, because I've definitely
think it was noticeable.
Yeah.
Yeah.
I mean, they definitely they both in and out of long day and short day season, we call them, still have a glycogen body for sure.
Very cool.
Yeah. Good question. It's like,
That's a
I
great
feel like
question.
it was a convenient answer. I mean, like happenstance. I wouldn't have thought that's good.
Is there anything about the particular control problem that they're solving that suggests any potential type of signal that would need to be sent? This is kind of on the back of what Ben was asking before, like, yeah. Oh, there you go.
We have made some attempt to answer that question. This is another project that's it's like in progress, and I think some version of it Katie is in the middle of trying to write up in some capacity or another. And so what Katie did in collaboration with a graduate student, Max, and also Steve, is try to look at it from a controlled dynamical systems perspective. I'll just introduce the model, and Katie knows way more about all the details. So what we decided to do was ask this question as a why question, like why would you have such a thing? And what is it good for in terms of observability and controllability? And so what Katie and Max did was take a now classic spring-loaded inverted pendulum model, which has been used a lot for studying bipedal walking. So it's basically a mass on a spring-loaded stick. So instead of just being an inverted pendulum, it's an inverted spring-loaded pendulum, okay? And then what we did is we added another one on top because one is not enough. So now it's a spring-loaded inverted double pendulum. And that is the minimum number you need to stick a beak on it to try to solve the head stabilization problem. So if you consider the top ball to be the head, what do you have to do with your midsection to stabilize the head if the ground is moving? And what if we play these games about putting different numbers and different types of sensors in either the head or the midsection? The slide is a bit old, so don't take any of the diagrams too literally. There's been many iterations of this since, but that's sort of there's another set of projects that we've gone partway down to try to answer this question.
Was there any temptation to use one of those birds that tips down and drinks the water, so you could use Simpsons GIFs in your talks?
I know, right?
That's
Yeah,
a good. That's a great idea, and I'm definitely doing that at some point. I know exactly what you're talking about. So I will say where we got kind of hung up here. So I have two thoughts now that being brought up. What was thinking about how those sensors are inputs to a state space system, and you know, and how that becomes a good model of the bird. You know, is it a good model of the bird or not, of what's actually happening, right? So as an acceleration sensor, we don't just want to, if our states are position and velocity, we don't just want to integrate our acceleration sensor to make it a velocity input. That's not really the question we're asking. The other thing that I think it was Ben or Eli brought up, but the idea of which direction gives you the best signal is really a rigid body problem and not a point mass one. Mm-hmm. So we might be, so that's, I mean, it's something that we definitely came up before. Obviously we started here for simplicity, but of even just the slip model, but with the rigid body on top is quite interesting. The system. Yeah, but then that requires your 3 degree, your 6, or at least your 3 rotational degrees of freedom. The math gets annoying.
The math gets kind of hairy.
Yeah.
One of the things I was thinking about a little bit when I was watching the first videos you showed, Bing, was this kind of idea of like if I'm grounded to the world, I could like, you know, like make one of my arms floppy and it wouldn't really matter if people were like smacking it around, it wouldn't affect my body. But the curious thing about the bird is that it's not grounded at all, and yet it keeps its head still. And so I wondered if that having the bird sort of standing on the pendulum was sort of giving it the stability of the ground for free. And the curious part is that the head is like rigid still, and this whole thing can move around, but it can't like tether to anything. It's not like there's an ether it can bind to or something.
Yeah. So the idea is that eventually to apply like an input force or an input signal to the feet, like a branch bobbing up and down or something.
Yeah. Okay. Yeah. Cool.
No, it's a good point.
Well, sounds like we may be at the end of our questions. I think we have likely left you with more questions than we have answers. But that's okay. That's kind of where we've been for a while. Thank you for your interest. This
Thanks
is
so much
obviously
for
something
coming.
we think about all the time, and we love